So let's talk about the epididymis. So the point was that after exactly the production of the sperm in the seminiferous tubules, we're going to have the movement from the seminiferous tubules to the straight tubules, then to the rete testis, and then exactly from the through to the head of the epididymis through the afferent ductules. Now, of course, the point is that all of these tubes have in fact different epithelium and different characteristics. Specifically, we're going to see that we have a different movement from the Sertoli cells, exactly from the seminiferous tubules, toward cuboidal, toward columnar, toward pseudostratified columnar cells. The point is that we're going to have a very, very peculiar epithelium in the afferent ductules. We're going to have a mixture of cuboidal cells that do not have exactly villi at all, and we're going to have mixtures of tall cells that in fact have stereocilia. So this is the very, very peculiar, let's say, characteristic of this epithelium, and this exact differences can be seen in this very, very useful table that practically classify the differences of the duct system from the male genital ducts. Now, as we saw in the seminiferous, we're going to have typically the Sertoli cells, and that's it, of course, in the whole mucosal layer, aside, of course, the spermatogonia. In the straight tubules, we're going to find again the Sertoli cells with simple cuboidal. Then we're going to go to rete testis, still cuboidal, and here we find this very unique epithelium, which is in the afferent ductules of the simple cuboidal, non-ciliated. And in this case, we're going to have the simple columnar ciliated structures. Exactly after the afferent ducts, we're going to have the observation of stereocilia in the epididymal duct. In fact, the stereocilia is going to be one classic giveaway that this duct you're looking at is 100% sure the epididymis. Because of course this is a very typical characteristic of the epididymis. What is important to notice is in fact that the length of stereocilia, this is the biggest in size, the biggest in length, of course, the biggest in size apical specification and specialization of the cells of the epithelium. In fact, this is going to be your giveaway, and you should always be cautious to see it. And in fact, it's very, very easy to notice it because of its very, very big length. Now, the other differences we should start seeing, of course, is that the support exactly what is exactly beneath the mucosa. Now, we saw that at the beginning, the seminiferous tubules, we're going to have the myoid cells and the loose connective tissue. And interspersed within the loose connective tissue, we're going to find, of course, the Leydig cells. The straight tubules are going to have only a very thin line of the connective tissue. Rete testis is going to have a dense, irregular connective tissue. And now here starts the differentiation. We're going to start seeing the presence of smooth muscles. Now, why is that? Because the point and the function of the epididymis is to absorb, one of the functions is to absorb water. Why would we need that? Well, take a look at the anatomy of the epididymis. So we do know that the only movement that allows the sperm to go from the seminiferous tubule to the epididymis is what? Just the myoid cell, and that's it, and very, very short amount of smooth muscles. This is not enough by itself. Weak contractions are not enough and to sustain the whole movement of sperm from the seminiferous tubules to the head of the epididymis. But we forget one basic characteristic: this absorption of water. This is exactly the function of stereocilia. The stereocilia is an absorptive specialization. So because we have in the epididymis a lot of cells, a lot of tall cells, pseudostratified cells that actually continuously absorb water, now this creates a fluid flow of the cells of the sperms from the seminiferous tubules to the head of the epididymis. So the biggest force that drives the movement of sperms from the seminiferous tubule towards the head of the epididymis is exactly this absorptive, this fluid flow from one point to the other. Now, interestingly, the head of the epididymis and the body contain only one layer of smooth cells. Why so much? What's the point? Why do I specify head and body? And in comparison, the tail has 3 layers of the smooth muscle cells. Well, because the primarily force, the movement, the mechanical force that drives the movement in the head and body is this absorptive, is this fluid flow. And in comparison, the tail actually needs to have a more let's say, wider, more prominent smooth muscle layers in order to facilitate this movement from the tail to the vas deferens, to the Ductus Deferens or vas deferens. So this is exactly the structure and the point of the epididymis. Of course, epididymis also plays a role into the maturation of the acrosome. Here we're going to have the decapacitation process that in fact we're going to see the inactivation of the acrosome, and of course this is a stage that allows the acrosome to work properly when it reaches the zona pellucida, in this case of the in the case of ejaculation. So in this case, this is the two functions of epididymis: storage of the sperm, maturation of the sperm, and of course movement and contraction with the movement of the sperm through the whole body and head, body, and tail of epididymis in the case of the ejaculation. So let's actually visualize the structure with the microscope. Now, commonly speaking, you should see the section that contains the testis along the epididymis on exactly the same slide because of the different sections. Now, again, they give away the straight characteristic. As soon as you see, you'll know that this is the epididymis, is going to be the stereocillia. I have only one more location of stereocillia in the human body, and this is in the inner ear. And of course, the morphology there is completely different. So let's zoom in. So when we zoom in, I can actually see the very, very long stereocilia on the top of the pseudostratified epithelium right here. And the point, of course, again, is to absorb fluid that is being produced by the Sertoli cells. And this is exactly the last feature of the absorption from the Sertoli cells. So exactly, this is the very typical characteristic morphology of the very, very prominent stereocilia with a very, very tall pseudostratified epithelium in this case. And of course, around it, we're going to find the connective tissue structures, and of course, this one layer of smooth muscles. And in fact, here we can see the very, very clear presence of the smooth muscle based, of course, on the S-shaped contraction of also the nucleus in this case of the whole cell, and as a consequence of the nucleus. So let's move on. So let's talk about the vas deferens, also called Ductus Deferens. Now, as the name suggests, it's a very simple function. This is a duct that allows the movement of sperm from the vas deferens to the ejaculatory ducts. So again, the morphology is very, very simple. We're going to have the typical pseudostratified epithelium in the middle, in the center, and the lumen, of course, of the vas deferens. Exactly, so we're going to find the smooth muscle cells, the lamina propria, and then exactly the smooth muscle cells. Importantly, the one important feature here is that we're going to find three different layers of smooth muscles. First, the inner longitudinal, the middle circular, and the outer longitudinal. You can see again exactly these features exactly right here in the same slide. Now, if we notice exactly the same thing in the histology guide, we're going to see exactly the same features. So again, the duct inside has the pseudostratified columnar epithelium that we already discussed. It's a very similar to classic epithelium with again the columnar cells, the basal cells exactly in the border and the close to the basal membrane. And exactly beneath, we're going to start seeing the three layers of the muscle. Now, this is the first inner longitudinal section. We can see this is a cross section, of course, of the smooth muscles. Now, this is the circular. We can see exactly the whole point, exactly the same feature, along with the very, very nice contractions of the smooth muscles. We can actually see it right here. This is a very, where is it? There is one typical, again, contractory state of the muscle, the smooth muscle in this case. And lastly, the outer lung, which has exactly the same morphology as the inner, as we saw before in the inner layer of the smooth muscles. Now, the whole point of the Ductus deferens is to move and to contract all these smooth muscles in many, many motions, similarly to the GIT contractions, the peristaltic movements. And of course, this allows the movement from sperm from the one point to the other. So let's move on. So let's talk about the accessory vesicles. Here, in fact, we're going to have the one very, very important vesicle, the seminal vesicle. Of course, we can see just by the picture of the anatomy here, this microscopic one, we can see that the ampulla is exactly the end point of the Ductus deferens, which is an opening. There's a complete opening and the dilation of the vas deferens. Now, again, the morphology is the same. The difference is that here we're going to have the convergence of the seminal vesicle with the ampulla to open up to the ejaculatory duct. Now, what is the function of the seminal vesicle? The function of seminal vesicle is actually to produce fructose and to produce glycoproteins in order to start creating and forming the fluid of semen. This is the first stage of the formation of semen. So the whole point here is that actually the seminal vesicle produces fructose, which is going to be the source, the energy source for the sperm cells, in order to be able to move and to reach the point. And now here in this section, we're going to start seeing exactly the ampulla of the vas deferens, and of course the seminal vesicle right here. So or exactly we're going to see the morphology that is similar. We're going to see the mucosa right here and exactly beneath the smooth muscle cells exactly beneath. Now, important to notice that this is the seminal vesicle produces 70% of the ejaculate, makes up the whole, like the 70% of the semen that we know it. And of course, it contains glucose. I told you before that this is the source, the energy source for the sperm cells. The PG, the prostaglandins, are of course molecules that play a big role in the facilitation and allow the whole process of fertilization exactly in the female reproductive tract. And lastly, the fibrinogen, which is the component that is responsible that makes the semen coagulate. So exactly, let's see this structure again in the microscope. So in this section, we're going to find exactly both structures of the vas deferens and the seminal vesicle. So if we zoom in the seminal vesicle, we're going to start seeing the mucosa. Even by this magnification, we can see that this area is highly convoluted and we're going to have many folds. So by zooming in this area, we're going to start seeing again the typical pseudostratified features. Unfortunately, because of this high convolutions, this high, let's say, glomerular structure in this case, we're going to have the not easily discernible tall pseudostratified columnar epithelium, and exactly beneath we're going to start seeing again the basal membrane. Attached on them we're going to find as always the basal cells. Beneath that we're going to start seeing the two different layers of smooth muscles, the inner circular and the outer longitudinal section. Of course, here again the contraction of the smooth muscles allow the seminal fluid to process and to actually move out to the ejaculatory duct, as we can see exactly in the same picture. So this is again the ending point of this contracting event. Now the next vessel we're going to discuss is going to be the prostate. Now the prostate has a distinct function, which is to produce mucus. Now we're going to start seeing the three different areas of the prostate. We're going to first see the transitional zone, then specifically the central zone, and lastly the peripheral zone. This is the 3D structure, of course, of the prostate. When we have this when we have the section of the structure, we're going to start seeing overlapping terms, and it's not that easy to distinguish, of course, the different areas. But first, let's talk about the functions of the different zones. Well, the first zone we're going to have is the transition zone, which is the innermost part, as we can see the picture exactly right here. Sorry, this is the transitional zone, and this is of course going to be 5%. We're going to have the mucosal glands that here are going to be the 5% of the whole population of the peripheral mucosal glands. Now, of course, this mucus is going to be clear mucus. It's not going to be the typical mucus that we know, for example, in the nose or the GIT tract. The exact zone we're going to have the central zone. We're going to find here the submucosal glands with a bit longer ducts. The point, there's no major difference in the specific function of the different areas. And last, we're going to have the peripheral zone that have the 70%. This is the longest amount of duct, and of course, this is the biggest area of the whole section. Now, so before we actually see the picture in the microscope, we should start noticing the epithelium right here. This is going to be cuboidal. And there are going to be areas that are going to have cuboidal epithelium. There are going to be areas that are going to have columnar epithelium. So this is a mixture, and it's not that actually clear in many sections which is which. So first we can see exactly right here the glandular epithelium. This is the mucosa again that contains what I just told you before, the semicolumnar epithelium, and there are regions of pseudostratified. Now there are not that many in this section, and I told you it's not that easy to discern where we have the overlap and where we have the actual presence of the clear columnar and the clear pseudostratified epithelium. So it's not that easy in these cases. The question is, how do I identify this picture, this structure in the microscope when we see it? There's going to be one unique feature. You see exactly these bodies, these very huge, let's say, we can call them aggregations of content. Specifically here, this is going to be called the corpora amylacea. Now these are going to have very many different components, such as, for example, keratan sulfate and other components. This is a natural structure. This is a physiological structure. We can see this formation of the corpora amylacea in older as men grow. So this based by age, based on the age, we're going to have more and bigger in size corpora amylacea. Now these are concentric cells of lamellar appearance. So this is exactly the structure. That is the basic and dead giveaway that this is going to be the prostate in the section that we have exactly right here. Important to notice that this exact corpora amylacea contains more structures such as glucoproteins. And this is again this is a more typical picture and appearance of the corpora amylacea. So to sum up, we have the production of the sperm in the testis. Now the convergence of the seminiferous tubules towards the epididymis, which of course the epididymis stores and matures the sperm. After the movement through the fluid flow of the absorption of the epididymis absorbs fluid continuously and as a consequence drives the sperm from the testis to the epididymis, where it's again being stored and maturated. Of course, there we're going to find the decapacitation process, which is of course which assures the inactivation and the not ectopic activation of the acrosomal cap. It's very, very important, of the acrosome in this case. Then we're going to have the movement of the epididymis from the epididymis towards the Ductus Deferens that allows the movement of sperm from the epididymis to the ejaculatory duct. Of course, here we're going to find that the tail of the epididymis is going to have the same smooth muscles layers, again the longitudinal, circular, and again longitudinal, that allow for this peristaltic motion that allow and facilitate the movement of the sperm from the epididymis toward the ejaculatory duct. Then we're going to start seeing the seminal vesicle. This is again the first area we're going to have the production of fructose, prostaglandins, and most important, and also very, very importantly, fibrinogen that makes actually the sperm and semen coalesce. Then we're going to have the next prostate gland. The prostate gland, in fact, is the area where we're going to have the production of clear mucus in specific area. And lastly, we're going to have the bulbourethral gland. We didn't discuss this, and this is not good. This is a very, very short topic. You should just see once in Junqueira, and that's it. Now, here again, we have the production of clear mucus again in the bulbourethral gland that practically allows the easier movement of semen from through the whole structure of the penis. Thank you very much.